Nature does not make foldable proteins by accident. The ratio of stable protein folds to random sequences is 1 in 10 to the 77th power. To put that in perspective, that's a number with 77 zeros after it, more than the number of atoms in the observable universe. Yet AlphaFold has learned to predict these astronomical odds with 90% accuracy. How is that possible? And what does it tell us about the deepest secrets of life itself? Welcome to Mindcast. I'm Will, and today we're diving into what might be the most profound scientific convergence of our time, a story that connects artificial intelligence, ancient biochemistry, and the search for alien life in ways that will fundamentally change how you think about existence itself. We're exploring breakthrough research called the architecture of Genesis, and I promise you, by the end of this episode, you'll understand why 3 seemingly unrelated scientific revolutions have converged to unlock the universal grammar of life. Here's what we're dealing with: the convergence of 3 revolutionary frameworks. First, the amyloid world hypothesis, a radical reimagining of how life began that challenges everything we thought we knew. Second, AlphaFold, Google's AI that has essentially mapped the entire universe of possible proteins. And third, assembly theory, a new physics-based way to detect life anywhere in the cosmos without assuming it looks anything like life on Earth. What makes this convergence so extraordinary is that these 3 breakthroughs don't just complement each other. They reveal that the secrets of life's origins are encoded in the fundamental laws of physics. We're not just talking about how life began on Earth. We're talking about a universal biology that could help us recognize life anywhere it might exist. Let's start with a revolution that's been brewing in origin of life research, one that completely overturns the textbook story you probably learned in school. Our first key insight is what I'm calling the amyloid revolution, and it starts with destroying one of biology's most beloved theories. For over 50 years, scientists have believed in something called the RNA world. The idea was elegant. RNA could both store genetic information like DNA and catalyze chemical reactions like proteins. It seemed like the perfect first molecule of life, a Swiss Army knife that could do everything needed to jumpstart biology. But here's the devastating problem. RNA is incredibly difficult to make under realistic early Earth conditions. The building blocks of RNA, nucleotides, are like trying to assemble a Swiss watch while riding a roller coaster in a thunderstorm. Ribose, the sugar backbone of RNA, literally falls apart in water over geological timescales. Even worse, linking these nucleotides into long, stable chains without sophisticated enzymes is thermodynamically unfavorable. The RNA world required a level of chemical sophistication that the chaotic early Earth simply couldn't provide. Enter the amyloid world hypothesis, and this is where things get fascinating and a little unsettling. Instead of complex RNA, what if life started with something much simpler? Short chains of amino acids called peptides. Now, when most people hear amyloid, they think of disease, Alzheimer's plaques, protein tangles destroying brain cells. But here's the revolutionary insight that's rewriting the origin story of life. Those same molecular structures that cause neurodegenerative diseases might have been the engines of creation 4 billion years ago. Think about the elegance of this solution. Amino acids are everywhere in the universe. They form readily in Miller-Urey experiments. They rain down on meteorites. They're probably sitting in the oceans of Europa and Enceladus right now. The barrier to entry for a peptide world is incredibly low compared to an RNA world. But the real magic happens when these simple peptides start to self-organize through pure physics. Here's where thermodynamics becomes the hero of our story. Short peptides have an innate, thermodynamically driven urge to stack themselves into incredibly stable structures called amyloid fibrils. Picture molecular Lego bricks that desperately want to click together. These structures form what researchers call cross-beta sheets, stacks of peptides running perpendicular to the fibril axis, and once they start forming, they become virtually indestructible. This is crucial. Amyloids are thermodynamic black holes. They represent the most stable configuration a polypeptide chain can adopt, what physicists call the ground state. In modern biology, proteins fight against this collapse, spending enormous energy to maintain their functional but less stable shapes. But in the harsh, chaotic environment of early Earth, this hyperstability was exactly what was needed. While more delicate molecules were being destroyed by heat, radiation, and chemical chaos, amyloids survived and thrived. But here's where the amyloid world gets truly mind-bending. These structures don't just persist, they replicate. Imagine a growing amyloid fibril made of left-handed amino acids floating in a primordial soup that contains both left and right-handed amino acids in equal measure. Due to simple physical geometry and what chemists call steric hindrance, the growing amyloid can only recruit other left-handed amino acids. It literally acts as a chiral filter, rejecting right-handed amino acids because they simply don't fit the geometric pattern. This solves one of biology's deepest mysteries: why all life on Earth uses left-handed amino acids when abiotic chemistry produces both types equally. The amyloid acts as a template, propagating its own structure and forcing new matter to conform to its pattern. This is template-directed replication that predates DNA by billions of years. The information isn't stored in a genetic code, it's stored in the physical shape itself, and this mechanism is identical to how prions work today, suggesting that those infectious proteins aren't aberrations but ancient echoes of our pre-genetic past. This brings us to our second revolutionary insight: how an artificial intelligence accidentally became the ultimate decoder ring for the language of life itself. In 2021, Google's DeepMind released AlphaFold 2, and it didn't just solve the protein folding problem, it obliterated a 50-year-old scientific barrier that many thought might be fundamentally unsolvable. The problem AlphaFold solved was called the Leventhal paradox. Here's the issue. A protein chain has so many possible ways to fold, so many degrees of freedom, that if it tried them all randomly, it would take longer than the age of the universe to find the correct shape. Yet proteins in your body fold correctly in milliseconds. How? The answer required AlphaFold to become an evolutionary detective. Instead of simulating physics atom by atom, which would be computationally impossible, AlphaFold uses pattern recognition grounded in evolution. It takes a protein sequence and searches databases for evolutionary cousins, looking for what researchers call co-evolutionary signals. If amino acid 50 and amino acid 100 always change together across different species, if one gets bigger, the other gets smaller to make room, the AI infers they must be touching in the final 3D structure. It's like reconstructing a crime scene by interviewing all the witnesses simultaneously. But here's where AlphaFold becomes truly revolutionary for understanding life itself. It didn't just predict individual proteins, it mapped the entire protein universe. We went from knowing the shape of about 180,000 experimentally determined protein structures to over 200 million predicted structures in a single leap. And when scientists analyzed this massive dataset, something profound emerged. The number of possible protein shapes isn't infinite. It's finite and surprisingly small. AlphaFold's predictions cluster into a discrete number of fold families, revealing that the geometric solutions to packing a chain in 3D space are fundamentally limited. Think about what this means. Form in biology isn't arbitrary. Just as planets must be spherical because of gravity, proteins must adopt specific architectures, helices, barrels, sheets, because of the immutable laws of atomic physics and geometry. This finite fold space suggests something incredible about convergent evolution extending to the molecular level. If alien life uses similar chemistry, they'll likely discover the same protein shapes we have, the same helices, the same barrels, the same sheets. It's not just that life might be universal. The actual molecular machinery of life might follow universal patterns dictated by physics itself. AlphaFold also illuminated what scientists call the dark proteome, protein sequences that had no similarity to anything we'd seen before. These were the dark matter of biology, ubiquitous but mysterious. Before AlphaFold, they were just strings of letters with unknown functions. Now we could see their shapes, and some were completely unique, orphan folds that exist nowhere else in our databases. Some might be living fossils from the earliest days of life, ancient architectures lost in the lineages that led to modern cells. But here's the crucial limitation for astrobiology. AlphaFold 2 relied heavily on evolutionary history. If a protein had no evolutionary cousins, if it was a true orphan or an alien molecule, AlphaFold struggled. However, AlphaFold 3 changed everything by learning physics directly from atomic coordinates, moving away from evolutionary dependence toward what researchers call a universal folder, a system that could potentially fold anything, even alien proteins we've never seen before. This brings us to our third revolutionary insight. How do we actually detect alien life without falling into the trap of looking for Earth-like biology? Enter assembly theory, a framework that's going to fundamentally change how we search for life in the universe. The biggest risk in searching for extraterrestrial life is what researchers call Earth bias, looking for DNA, water, carbon, all the specific details of how life works here. But what if we're thinking about this completely wrong? What if life isn't a substance, but a process? What if life is fundamentally about information processing, evolution, and the generation of complexity, regardless of the specific chemistry involved? Assembly theory provides a substrate-independent way to recognize this process. Here's the core insight. The universe doesn't create complex objects in high abundance by random chance. Complexity requires what physicists call history, a sequence of selection steps that builds complexity incrementally. Assembly theory calculates something called the molecular assembly index, the minimum number of steps needed to build a molecule from basic building blocks. The magic threshold is around 15 assembly steps. Simple molecules like water, carbon dioxide, and basic amino acids score below 15. These can form naturally in stellar nurseries, hydrothermal vents, or other abiotic environments. But complex molecules like the cancer drug Taxol or the nucleotide adenosine score well above 15. If you find molecules with high assembly indices in abundance, they must be the product of an evolutionary system, either biological or technological. Now here's where AlphaFold and assembly theory create an incredibly powerful synergy for detecting alien life. Imagine a space probe on Enceladus detects a high-complexity polymer in the subsurface ocean. Assembly theory flags it as interesting because of its high assembly index. We then feed that polymer sequence into AlphaFold. If AlphaFold predicts the molecule folds into a stable, compact structure, a barrel, a helix bundle, something that clearly has functional architecture, that's our smoking gun. Because remember that impossible ratio I mentioned at the beginning, 1 in 10 to the 77th? Nature doesn't make foldable proteins by accident. Finding a molecule that both has high assembly complexity and folds into a stable thermodynamic structure is mathematical proof that an evolutionary process has been grooming that sequence to perform a function. This is the essence of what researchers are calling. The implications are staggering. Scientists are already designing missions equipped with what they call the Darwin probe, a three-part system that could revolutionize astrobiology. First, a mass spectrometer calculates assembly indexes in real time, filtering out simple molecules and targeting the complex ones. Second, a nanopore sequencer reads the sequences of these complex molecules. Third, an AI core runs AlphaFold to test if these molecules fold into stable, functional structures. Let me synthesize these revolutionary insights and give you 3 concrete takeaways that could reshape how we understand our place in the universe. First takeaway: life isn't fighting against physics, it's the inevitable results of physics. The amyloid structures that cause disease in your brain today are the same structures that sparked life 4 billion years ago. Every protein in your body is locked in a constant battle against what researchers call amyloid gravity, the thermodynamic pull toward the most stable configuration. Your cells spend enormous energy to prevent this collapse, but when they fail, disease results. This isn't a design flaw, it's a direct connection to life's thermodynamic origins. Second takeaway, we're witnessing the birth of computational astrobiology. By training AI systems on the fundamental physics of protein folding, we can simulate alien biochemistries before we even discover them. We could model silicon-based proteins, ammonia solvent biochemistry, or life based on entirely different chemical alphabets. We're building a reference library of potential alien biosignatures, preparing for discoveries that haven't happened yet. When we do encounter alien life, we'll be ready to recognize it. Third takeaway: the search for alien life is becoming a pattern recognition problem. Instead of looking for specific Earth-like signatures, we're learning to recognize the mathematical fingerprints that any evolutionary process must leave behind. Life is the mechanism that explores vast chemical space and finds the tiny islands of sequences that fold and function. AlphaFold has learned the map of those islands, giving us a universal compass for recognizing organized complexity wherever it might arise. Here's what keeps me awake at night thinking about all this. We're standing at an incredible threshold in human understanding. The convergence of amyloid chemistry, artificial intelligence, and assembly theory isn't just giving us better scientific tools, it's revealing that there's a universal grammar underlying all possible biology. We're moving from asking, "What is life?" to "What are the fundamental constraints that any life must obey?" And those constraints, thermodynamics, geometry, information theory, are the same everywhere in the universe. When we do find alien life, it won't speak English and it might not use DNA, but it will almost certainly obey the laws of protein folding that AlphaFold has learned to recognize. The research we've discussed today represents a fundamental shift in biology, from cataloging Earth-specific creatures to understanding the theoretical physics of life itself. We're not just studying what happened to evolve here, we're mapping what's possible to evolve anywhere. We're discovering that the universe has a vocabulary for life, written in the language of molecular geometry and thermodynamic inevitability. I want to leave you with this thought. Every time you move your hand, you're witnessing billions of proteins choosing their shapes according to 4 billion-year-old rules. Those rules aren't accidents of terrestrial history, they're fundamental laws of how matter organizes itself when it wants to be alive. And now, for the first time in human history, we understand those laws well enough to listen for them in the silence of space and recognize the voice of life when we hear it. That's it for today's Mindcast. If this episode sparked something for you, I'd love to hear about it. Share it with someone who's curious about the deepest questions of existence. And remember, we're living through a moment when artificial intelligence, ancient chemistry, and space exploration are converging to answer the ultimate question: are we alone? The tools to find out are being built right now, and the answer might be closer than we think. Until next time, keep questioning everything.